Hey guys, welcome to our very first lecture of A&P 2. We are going to start right where we left off last semester with our finishing up of the nervous system. So our first week is dedicated to the special senses. We have 5 different special senses: olfaction, which is the sense of smell; gustation, which is your sense of taste; and then vision, equilibrium, and hearing. We're going to begin with smell, or olfaction. And so olfaction, we have olfactory organs, which are what provide us our sense of smell. And you find those in the nasal cavity on either side of the nasal septum. So you've got olfactory organs on both sides of the internal nares. There are 2 different layers that make up the olfactory organs, which are the olfactory epithelium and the lamina propria. So we'll look at these each separately. The olfactory epithelium contains olfactory receptors, supporting cells, and stem cells. It covers the cribriform plate, the perpendicular plate of the ethmoid bone, and the superior nasal conch. So that whole upper region is covered by this olfactory epithelium. Deep to the epithelium, we find the lamina propria, which contains olfactory glands, also called Bowman's glands, and those secrete a secretion that, when combined with water, forms the mucus that we know to find within our nasal cavity. So it's important to remember that the Bowman's glands, or the olfactory glands, are actually found within the lamina propria, and you can see it right here that gland is in the lamina propria, but then the outlet, or where the mucus production exits, is on the tip edge of the epithelium, but the gland itself is located within the lamina propria. There's also areolar tissue, which is pretty standard in lamina propria, blood vessels, and nerves that are going to supply the region. So the secretions from the olfactory glands are going to coat the surfaces of that epithelium. And then the olfactory receptors, which are highly modified neurons, you can see down here that those receptors end in what are called dendritic bulbs. And those dendritic bulbs are the branched tips of the dendrites that are actually projecting through the epithelium and then lying within that mucus. And those are going to be detecting odorants, which are the odors, the molecules that are dissolved within the air. So as you inhale, those odorants are going to bind to those odorant binding proteins within the dendritic bulbs of the olfactory receptors. When you inhale air, you're detecting about 2% of the inhaled air when you're smelling for odorants. And the more you inhale, the more exposure those receptors are going to have to additional odorants, so you'll be able to smell better when you inhale more and more air. So we're going to be talking about pathways today, and pathways are what happens with the impulses of the nervous system. So where does it start at the receptor, and where does it end? So the olfactory pathway begins at those dendritic bulbs in that epithelial layer. And then the axons from those neurons leave the olfactory epithelium, penetrate up through the cribriform plate of the ethmoid bone. Remember, we talked about that in A&P1, where if you were to somebody were to punch you really hard in the nose and you were to break your ethmoid bone, you could sever those axons and you could lose your sense of smell. So they leave the olfactory epithelium, they pierce through the cribriform plate, where they reach the olfactory bulbs of the cerebrum. And that's where your first synapse occurs. So it's one neuron going from the olfactory epithelium all the way up to the cerebrum. And olfaction is the only special sense that directly reaches the cerebral cortex. The rest of them all have to go through the medulla or other regions where they can be processed prior to being sent on to the cerebrum. So olfaction is special in that sense. So our first synapse occurs within the olfactory bulbs, and then the axons of the next neuron leave the olfactory bulb and travel along the olfactory tract to reach the olfactory cortex, the hypothalamus, and portions of the limbic system. So the link to the hypothalamus and the limbic system is what gives you, if you've ever smelled something and had this like really severe memory or reaction to the smell on like a gut level, that's because of the link to the hypothalamus and the limbic system. There was this soap that smelled like my dad, and a few years after my dad passed, I was at some craft fair and I was smelling different soaps, and I smelled this one, and it was like I immediately burst into tears because it just smelled like my dad. So it's really interesting how the olfactory tract can really trigger memories. Olfactory discrimination is the fact that we can distinguish between thousands of different chemical stimuli. And the central nervous system is going to interpret smells by the pattern of receptor activity, which is pretty cool. Humans can detect between 2,000 and 4,000 stimuli, but dogs can detect between 10,000 and 100,000 times better than a human can. So it's pretty incredible what dogs can smell. And animals in general, they have a much better sense of smell, which is very protective for them when they're out in the wild. One of the other cool things about our olfactory system is that the basal cells in the epithelium actually differentiate into new receptor cells. So the receptor cells have a pretty high turnover rate, and so as the basal cells, they grow and they differentiate and they become new neurons. So this is pretty rare to find within the body, but it happens within the olfactory epithelium. The number of receptors does decline with age, which is part of the reason why as you age, you lose your ability to smell as acutely or as well as you did when you were younger. So that was the end of our first sense. Our second sense that we're going to talk about is taste. And if you're hoping this is going to be a short lecture because that was short, hold on, because we're going to talk about vision for a really long time. So not to get too excited there for you. So sense of taste or gustation. Gustation provides information about the foods that you eat and the liquids that you drink. Our taste receptors, which are our gustatory receptors, are found on the tongue and then portions of the pharynx and the larynx, and they're found clustered into taste buds. Most people have heard of taste buds. It's sort of one of those things that are commonly spoken about, but we have multiple different types of taste buds. This should say 4 types, where it says 3 here, it should say 4. So we have 4 types of lingual papillae. Those are projections on the tongue. So our filiform papillae are for friction, and they do not contain taste buds. So they're just the raised bumps on the tongue, and those are for friction to sort of grab onto your food, help move it around in your mouth. And we find those on the anterior 2/3 of the tongue, the filiform papillae. The fungiform papillae each contain 5 taste buds, and we find those on the tip of the tongue and the sides of the tongue. The circumvallate or just vallate papillae each contain 100 taste buds, and those are found in an inverted V shape along the back of the tongue. So you can see right here, those are the vallate papillae. And then the last kind are called foliate, F-O-L-I-A-T-E, and we find those on the lateral margins of the tongue with taste buds embedded within the folds of those foliate papillae. So if you were to look at the side of the tongue, you could see little folds in there, and so the taste buds in there are the foliate papillae, or papilla, however you would like to pronounce it. Our taste buds contain basal cells, which are stem cells that actually produce daughter cells that can mature into new gustatory cells. Gustatory cells are the cells that actually provide our sense of taste. So it's similar to olfaction, where we have those stem cells that can differentiate into new taste cells. And those gustatory cells have what's called a taste hair that are little microvilli that extend through a pore and then are able to detect the dissolved chemicals from food and drinks. So those are in the actual papillae. Each of those gustatory cells survives for about 10 days before they get replaced, so there's a lot of turnover again in the gustatory cells. Those cells are monitored by cranial nerves that synapse within the solitary nucleus of the medulla, and then from the medulla, information is sent on to the thalamus. And remember, the thalamus is like the great sensory collects all the sensory information and then sends on important information to the primary sensory cortex. So our cranial nerves for taste are cranial nerves 5, 7, 9, and 10. So the cranial nerve is going to pick up the information, send it to the solitary nucleus of the medulla, where it's starting to get processed. Then it gets sent on to the thalamus for additional processing. And then the thalamus is going to send it on to the primary sensory cortex, which is also the gustatory cortex of the insula. Interestingly, flavor, so if you're eating something and you want to say, what does it taste like? What is its flavor? Flavor is produced through a combination of taste and smell. So you taste things and then you smell them, and that combination produces what we think of as flavor. We have four primary taste sensations: sweet, salty, sour, and bitter, and then two additional sensations, umami and water. So umami is a pleasant, savory taste that we get through exposure to an amino acid called glutamate. You might have heard it if you ever were like a Iron Chef watcher back in the day. They talked about umami a lot. I don't know if they still do. I haven't watched in years. And then water, we have water sensors in the pharynx, and when they're stimulated, it decreases the release of ADH, or antidiuretic hormone, and therefore helps to regulate water balance and blood volume. So it's pretty cool that the nervous system automatically is sensing the amount of water that you're taking in to accommodate how much water you're going to be releasing out of your system. Gustatory discrimination, we discriminate dissolved chemicals through when they come in contact with our taste hairs. They're going to bind to receptor proteins of the gustatory cell and then create an action potential which goes along the pathway that we previously discussed. Ending in, so neurotransmitter released by the receptor cell is what's going to stimulate the next neuron in that pathway. The number of taste buds is going to decrease rapidly by age 50. So when you're young, you have a much more sort of distinguished palate, and over age 50, you start to change your tastes or not be able to taste as well. That also goes along with the decrease in olfactory receptors. We are about 1,000 times more sensitive to sour, which are acids, like acidic foods are considered sour. So 1,000 times more sensitive to sour than we are to sweet and salty. And we're about 100 times more sensitive to bitter than we are to sour. Now, these are important control mechanisms because bitter foods are often toxic and acidic foods can damage your GI tract. So we're much more sensitive to foods that can harm us than we are to foods that are maybe enjoyable. So we are most sensitive to bitter foods and then to acidic or sour foods, and then least sensitive to sweet and salty foods. We're going to spend quite a bit of time talking about the eyes and our sense of vision. So first thing we're going to do is look at the actual gross anatomy of the eye. So the eyes contain half of all sensory receptors in your human body. That's pretty incredible. If you think about, you have sensory receptors throughout all of your skin, through your all of your organs, and half of all of those receptors in your body are within your eyes. And it is the most complex of our special senses. So the functions of the eyes are light perception, so to perceive is it light or dark, color differentiation, so being able to tell what is red, what is blue, and then depth perception, being able to figure out how far something is away from you and being able to help you navigate your way through the world. The eye is an irregular sphere shaped. We have accessory structures called the ocular adnexa, and those are the eyebrows, the eyelids with eyelashes, the extraocular muscles, and the lacrimal system. And these all function to help protect our eye, lubricate our eye, and support our eye. The eyelids are also called the palpebrae. If you remember back to our muscular section, we learned about levator palpebrae superioris, which is the muscle that opens your eyelid. So the palpebral fissure is the separation between the upper eyelid and the lower eyelid. So when your eye is closed, the fissure is very small, and when your eye is open, it's very large, so it's at its largest. The medial and lateral canthus are what we see here in the corner of the eyes. So that's where those palpebrae come together on either side. The eyelashes are thick, robust hairs that help to prevent foreign objects from getting in your eye. So think about dust, anything like that that's going to get trapped by those eyelashes before it settles in your eye. And then tarsal glands, which are also called meibomian glands, secrete a lipid-rich or fatty product that helps keep your eyelids from sticking together. That product is called sebum. So they're little sebaceous glands, and those are found on the inner lining of the eyelid. The lacrimal system is the system of tear production, and that helps to lubricate your eye, prevent bacterial or viral infections, provide nutrients and oxygen to portions of the eye itself, and keep those surfaces moist. The lacrimal glands produce tears at the superolateral side, so the upper outer side of the eye is where you find the lacrimal gland, so you have one on either side. And the lacrimal gland produces about 1 milliliter of tears each day. The lacrimal gland is about the size and shape of an almond, and then it produces tears, and the tears drain down into the eye through the lacrimal ducts. And the lacrimal lake is in the base of the eye, so kind of down in this area. So if you can picture like you're about to cry, but you don't cry, and how you get tears that can kind of well up in your eyes, that is the area of the lacrimal lake. The punctum are the little pores at the medial surface of the eye, which drain the eye. So the lacrimal gland and the lacrimal ducts are out on the superior-lateral surface of the eye. And then so our tears are coming into the eye from the top, and then they're going to be leaving the eye from the medial and lower surface. So the punctum are little pores at the medial surface of the eye, which drain the tears down through canaliculi, which drain down to the lacrimal sac. And then the lacrimal sac drains through the nasolacrimal duct down into the nasal cavity. So additional tears from the eye get drained out and down into the nasal cavity. So you might notice when you cry a lot, your nose runs, and that is exactly because those tears are actually draining down into your nose. So looking at the eyeball, we've got 3 different layers that make up our eyeball. So the fibrous tunic is the outer layer, which is the tough, like sclerotic layer. The vascular tunic, which is the middle layer, which is also called the uvea or the choroid. And then the retina, which is the inner layer, which is also called the neural tunic. So tunic is another word for layer. So you have the fibrous tunic, the vascular tunic, and the retina. The fibrous tunic is the external layer. It's got the sclera, which covers most of the eyeball and is white in color, and then the cornea, which is transparent and covers the very front of the eyeball, and it's transparent so you can see through it. The fibrous tunic is exposed to the external environment, so it's much more durable than the other portions of the eye, which are protected by the sclera. So the sclera is made of dense fibrous connective tissue, and it helps to maintain the shape of the eye by resisting internal pressure and external forces. It also provides attachment for your extraocular muscles, so the medial rectus, lateral rectus, all the extraocular muscles that we learned about in A&P1, those all attach directly into the sclera, so to help move the eye. And the sclera is continuous with the cornea. So even though it looks very different, and the cornea is transparent, it is continuous with the sclera. The conjunctiva is a clear, moist mucous membrane that is non-keratinizing squamous epithelium. Remember, keratinized epithelium is dry, but we want our eyes to be moist, so this is non-keratinizing squamous epithelium. It covers the sclera and it lines the inside of the eyelids. It helps to lubricate the eye through mucus and tears and sort of holds on to that, and protects the eye by blocking the passage of foreign objects and any kind of microbes from outside deeper into the eye. The palpebral conjunctiva is on the inside of the eyelid, and the ocular and bulbar conjunctiva are on the outer surface of the eye itself. The cornea is a transparent fibrous coat that covers the anterior portion of the eye, including the iris, the pupil, and the anterior chamber of the eye. So it's basically covering all of this here. And the cornea is the first spot that light actually refracts. Refraction is the bending of light. So when light enters the cornea, it bends slightly, and then it goes through the lens where it's bent again to be focused later. So we're going to talk about that later, but I just wanted to introduce now that the cornea is the first point of refraction of light. The light that comes through the cornea goes to the anterior chamber and then through the lens and down into the eye. A corneal transplant is the most common transplant in the body, interestingly, and because there are no blood vessels in the cornea, there's no blood for your body to reject. So you never can reject a corneal transplant. So that's what makes them so common, which is great because being able to actually restore somebody's sight is an incredible gift. The cornea also obtains its oxygen through diffusion. Remember we said that the tears are bringing oxygen and nutrients, and they're washing the eye with it. So diffusion from the tears is going to bring oxygen into the cornea. The vascular tunic is the middle layer of the eye. It is pigmented with melanin, and it provides vascular is blood supply, right? So it provides the blood supply to the eye. So the blood from the vascular tunic also is going to diffuse nutrients and oxygen out into the sclera. The vascular tunic also forms the iris, which is responsible for your eye color. And the melanin reduces the reflection of stray light within the eye. So you don't want all kinds of light rays bouncing around within your eye because that's going to overactivate your photoreceptors. So when the light enters your eye, if it reflects, that reflection then is offset by the melanin that is found within the vascular tunic. The vascular tunic contains the choroid, the ciliary body, and the iris. So let's look at each of those. So the choroid is the name for the vascular structure within the vascular tunic. So there's lots of capillaries, little arteries and veins, as well as connective tissue within the choroid. It provides oxygen and nutrients to the outer layers of the retina. And this is where the melanin is contained. The ciliary body is a tissue that surrounds the lens. So you can see the ciliary body is here, and these ciliary processes are just those little processes right there, and they produce aqueous humor, which is a watery substance that fills this anterior chamber of the eye here. The ciliary muscles are contraction of those muscles alters the shape of the lens. So we alter the shape of the lens in order to focus what we're looking on. And so the ciliary muscles, the contraction of those muscles is what alters the shape of the lens. The iris is the colored part of your eye, and it surrounds the pupil, which is basically an opening within the iris. There's lots of blood vessels and pigment cells within the iris that give the eye its color. Eye color is determined genetically by the number of melanocytes within your iris, as well as the density of that pigmented epithelium. There are also 2 layers of smooth muscles within the iris, and they alter the size of the pupil. So we'll look at those in the next slide. The iris divides or separates the area between the lens and the cornea into an anterior chamber and a posterior chamber. And then the pigment within the iris is going to block light from passing through the iris into the retina. So light is only passing through the pupil. So here are those muscles that we were talking about. So the pupillary dilator, when it contracts, is going to open the pupil. As you can see here in this picture, that pupil is very dilated and open, letting in lots and lots of light. And then the pupillary constrictor is a sphincter muscle, and it's going to constrict the pupil down to almost like a pinpoint size. So depending on, if you remember when the very end of A&P1, we were talking about the sympathetic nervous system versus the parasympathetic nervous system. So with parasympathetic stimulation, where you are relaxed, your pupils are going to constrict because you don't need as much light coming in. And with sympathetic activation, where you are fight or flight, your pupils are going to dilate so that you can let in more light and see as well as you possibly can. So that is a pretty cool nervous system fun fact there. The retina is the inner layer of the eye, or the neural tunic. It's composed mostly of nervous tissue, so nerve fibers themselves, receptors, and the primary function of the retina is to form the image that you're looking at so that you can see what's actually out there. It's composed of a neural layer and a pigmented layer, and we're going to look at in great detail at all the different things that happen within the retina. So again, the retina is that inner layer of the eyeball, and it lines the posterior 2/3 of the inside of the eyeball. It constitutes the beginning of the visual pathway, and it can be viewed through what's called an ophthalmoscope, which is the scope that the doctor uses to look in your eye. And interestingly, the anterior surface of the retina is the only place in the body where the blood vessels can be viewed directly and examined for pathological changes. So like diabetic retinopathy, you can see changes in the blood vessels within the eyes, which A, is the leading cause of blindness in the United States, but also it's like a window into the body that we just don't have anywhere else. So it's really cool to be able to look in the eye and assess somebody's vascular health. The optic disc is this bright spot here, which is where the optic nerve, cranial nerve 2, exits the eyeball and heads back in toward the brain. We have pigmented epithelium, which is a sheet of melanin containing, it contains melanin. So it's another pigmented sheet that helps with decreasing bounce back of light within the eyeball. The neural portion is a multi-layered portion. So there are actually 2 layers to the neural portion. The outer layer contains the photoreceptors, and then the inner layer is additional neurons that begin to process visual information. So visual processing actually begins in the eyeball prior to triggering the optic nerve. And then we have the macula lutea, which you can see over here, and we're going to talk about that on the next slide. So the macula lutea is a small flat spot in the center position of the posterior portion of the retina. And the center of the macula lutea is what's called the fovea centralis, or the central fovea. And that is a small depression in the very center of the macula. Now, the fovea only contains cones, and cones are for color vision. So the center of the fovea is the highest visual acuity, or the sharpest vision of any area of the eye. So that's what you want to remember about the fovea, is it is the highest visual acuity, or the sharpest vision of the eye is within the fovea, and that it only contains cones. We're going to learn about rods and cones. And so it only contains cones in the fovea. Macular degeneration is another big cause of blindness in this country, and you can look at this link to show you some of the different things that can go wrong with your macula. Here's normal vision. So if you were to look at these 2 kids, a normal person would see both of their faces, but macular degeneration erodes the center of your vision. And so if you have macular degeneration, this is what it would look like. My grandfather had macular degeneration, so in order to see your face, he would look straight forward, and you would have to put your face over to the side of his eye so that he could see who you were, which was pretty interesting. So our photoreceptors, we have 2 different types, rods and cones. Rods are responsible for light and dark. So I always remember dark, D-A-R-K, rods. So rods are for dark. So vision within dim light are the rods. And then our cones are responsible for color vision. So cones, color, that's easy. Rods, dark, cones, color. And you can see this picture here that shows that they look a little bit different from one another. So our cones actually have this cone shape top, and our rods have more of a rectangular shaped top on them. In addition to color vision, your cones are also responsible for visual acuity. So how crisp of a picture do you see is based on your cones. So we have multiple different synapses within the eye. So the rods and cones are the photoreceptors. So you can see those here. Maybe I'll do this in red and make it thicker. All right, so here you can see your rods and cones. And those are going to synapse onto bipolar cells. The bipolar cells are going to synapse, so these will do bipolar cells in yellow. So here's our bipolar cells. And then our bipolar cells are going to synapse onto the ganglion cells. And the ganglion cells, the axons of the ganglion cells, all come together and create the optic nerve. So this is how our visual system, our pathway works, is we have rods and cones, bipolar cells, ganglion cells, and then the ganglion cells create the optic nerve. Between the rods and cones, or the photoreceptors, and the bipolar cells, there's a layer of horizontal cells. So you can see in here, there's one little cell right there that you can see that is a horizontal cell. And then between the bipolar cells and the ganglion cells, there's another layer of cells called amacrine cells. And those are right here, those amacrine cells. So what these are doing is just helping to integrate and coordinate all of the information that's getting sent. So they're sort of like an interneuron that are beginning that visual processing. So we have the rods and cones that synapse in the bipolar cells, and at those synapses we have horizontal cells. And then the bipolar cells synapse upon the ganglion cells, and along those synapses we have amacrine cells. And then the ganglion cells form the optic disc and then the optic nerve. So just to follow up, those horizontal cells and the amacrine cells, their purpose is to facilitate or inhibit the transmission of signals between the photoreceptors, the bipolar cells, and the ganglion cells, and therefore they have the ability to alter the sensitivity of the retina. So the optic disc is the portion where the optic nerve is going to leave the eye. So you can see, if I delete this, that we've got the pigmented epithelium back here. And then here are our layers of photoreceptors here, followed by the bipolar cells, followed by the ganglion cells. And the ganglion cells, all of those axons are going to come together and then turn and penetrate through that posterior wall of the eye to form the optic nerve. So at the optic nerve, where the optic nerve exits, we do not have, oops, I did not mean to do that. I'll just keep talking. Where the optic nerve exits, there are no photoreceptors, so that's where your blind spot is. So when I talk about a blind spot, I would encourage you all to do this with your book. It's really cool. I don't know if you can actually do it with your computer, but I bet you could. So you're going to use your right eye and look at the plus. So bring your face as close to the plus as you can. And as you pull your eye away from that plus sign, at some point that round dot is going to disappear. And that's when that dot is directly when the optic disc is the area of your eye that would be focused on that dot. And then you can focus your left eye onto the round dot, and as you bring it away, the plus sign will disappear. So this picture is in your textbook, and I would encourage you to play around with it. If you have kids, they love this. It's like incredible that something can just fully disappear off the page. So diabetic retinopathy, I just wanted to talk about that a second, is the blockage of small blood vessels within the retina. And once they get blocked, that then gets followed by the overgrowth of abnormal vessels within the retina. And all of that occurring damages the photoreceptors. Once the photoreceptors, because the photoreceptors require oxygen and nutrients, so when you've broken down those blood vessels, you are keeping the oxygen and nutrients from the photoreceptors, and so the photoreceptors begin to die, and that is why you end up losing your vision with diabetic retinopathy, for lack of oxygen and nutrients to the photoreceptors. So here we're just looking at that overall anatomy of the eye. So we've got the cornea on the front here. Let's see, we'll do this one. So here's our cornea, and then the rest of the eye is surrounded by the sclera, like that. So that's the exterior portions. And then we have an anterior cavity and a posterior cavity. So the anterior cavity is in front of the lens, and the posterior cavity is behind the lens. So the lens is the separation between the anterior cavity and the posterior cavity. However, in the anterior cavity, it's separated into an anterior chamber in front of the iris and a small posterior chamber behind the iris. So just keep that in mind, that we have an anterior cavity and a posterior cavity, but the anterior cavity is broken down into an anterior chamber in front of the iris and a posterior chamber behind the iris. And then the lens is the separation between the anterior cavity and the posterior cavity. So I kind of just went over that. Let's see, we have the anterior cavity, or the anterior and posterior chamber, separated by the iris. The cornea is the very outermost part of that anterior cavity. The anterior sclera, we have the iris within the anterior cavity, the ciliary body, the lens, and then that whole anterior cavity is full of that liquid aqueous humor. So the aqueous humor is a watery fluid. It's similar in composition to cerebrospinal fluid, and it fills that anterior cavity of the eye. It's secreted into the posterior chamber by those epithelial cells of the ciliary body, and then it flows through the pupil out into the anterior chamber. It's secreted at 1 to 2 milliliters per minute in the posterior chamber, and then it leaves the anterior chamber at the same rate. Its main functions are nutrient and waste transport, forming a cushion for the eye, so stabilizing the position of the retina, and that intraocular pressure, and helping to retain the shape of the eye. We measure intraocular pressure in the anterior chamber. So when you go to the eye doctor, they measure your pressure. And glaucoma is a disorder where you have increased pressure in the intraocular or within the eyeball, so increased intraocular pressure. There's a canal called the Canal of Schlemm, S-C-H-L-E-M-M, and that's the canal through which this aqueous humor is drained out of the anterior chamber. So if that canal gets blocked and the aqueous humor cannot drain appropriately, it's going to increase the pressure within your eye, and then that can begin to damage the retina and damage your ability to see. So having those pressures monitored is an important part of just general health of your eye. So here's that canal of Schlemm. It's the endothelium-lined tube in the eye that collects the aqueous humor and delivers it back into the bloodstream. And there's a little video that you can watch on glaucoma. The posterior cavity is behind the lens, and it includes the vitreous body and vitreous humor, which we're going to talk about next, as well as the retina, the choroid, and the posterior portion of the sclera. So the vitreous body is a gelatinous mass of vitreous humor. So aqueous humor is in the anterior cavity, so aqueous anterior. The vitreous humor or vitreous body are in the posterior cavity. So it is unlike the aqueous humor, which is continually being produced and drained, the vitreous body, it does not turn over. So you are born with it, forms during development, and it is not replaced. So it's very important to take care of your eyes so that they continue to function for you. The vitreous body is transparent, so you can see through it. It's colorless and it's gel-like, and so it fills up the space between the lens and the retina. So that whole open portion of the interior of your eye is full of this vitreous body. It also helps contribute to ocular pressure and maintaining the shape of the eye. And it holds the retina against the choroid. So retinal detachment can lead to a tear in the retina, which would damage photoreceptors and decrease your vision. So having the vitreous body hold that retina tight down against the choroid is very important. It also has a phagocytic function, so if you were to get some kind of microorganism within the eye, the vitreous body can help to clean that up before it ends up in an unchecked infection. And so here's a little video on retinal detachment that you can enjoy at your leisure. So the purpose of the eyes obviously is for vision. So related to vision, we have the eyeball, which is the globe of the eye, the optic nerve, which is the cranial nerve that goes from the eye to the brain, and then the brain, which is our processing center. So vision is achieved when cells of the eye are activated by light in the external environment. I think everybody should understand that already. So the lens is a primary structure involved in bending the light in order to focus light and produce a vision for you. So the primary focus of the lens is to focus the visual image on the photoreceptors by changing its shape. It bends the light or refracts the light. So in this picture here up on the top, you can see how light is coming in at one angle. It hits the cornea and gets bent for the first time at the cornea, and then it hits the lens and it gets bent a lot more to focus back on the retina. So remembering that the cornea is the initial spot of light refraction is important. This picture also shows you that the lens itself is a biconvex disc, meaning it is convexly shaped on either side. So it's not round, but it's more of like an oval shape. And it is held in place by the ciliary zonule, which are little suspensory ligaments from the ciliary body. And the ciliary muscles, when they contract, they alter the curvature of the lens. So in the top portion, you can see when the ciliary muscle is contracted, the lens is nice and round, and that helps when you're looking at something close up. And then when the ciliary muscle is relaxed, down in this second picture, you can see that the lens flattens out, and that's helpful for distant vision. So accommodation, this word accommodation down here is the, oops, my goodness, I shouldn't be allowed to touch stuff. Accommodation is the changing of the lens to accommodate between looking at something close up and looking at something far away. So it requires that lens to be nice and flexible. The lens is also clear. A cataract is damage to the fibers of the lens where the lens begins to become opaque and you can no longer see through it. So treatment for cataract removal really helps with visual loss due to cataracts. The lens is going to, when we're looking at refraction of light, again, it's bent first by the cornea, second by the lens. The focal point is the specific point where the intersection of the light rays occurs on the retina. So where you can focus most is where those light rays are going to be hitting the retina. And the focal distance is the distance between the center of the lens and the focal point. So that's just some vocabulary there. The focal distance is determined by the distance from the object to the lens and by the shape of the lens. So if it's round, it's going to be a short focal distance, and if it's flat, it's going to be a longer focal distance to help you, again, accommodate. And astigmatism is where light passing through the cornea and the lens is not properly refracted, so you get distortion of your visual image. Astigmatisms are very common and mild ones often go unnoticed, but a severe astigmatism can make it so that you can't see clearly, and then it would need to be corrected with corrective lenses. When we're checking vision, we look at visual acuity, so how clearly can you see? And so 20/20 is what we call normal vision. So that means when you're standing 20 feet away from the eye chart, you can see what the average person, and I'm putting average in air quotes, but what the average person can also see at 20 feet. 20/30 vision means that at 20 feet, you can see what a normal person would be able to see at 30 feet. So if a normal person was 30 feet away, they could see just as clearly as you can see at 20 feet. So that's 20/30 is having worse vision than normal. 20/15 vision means at 15 feet, you can see what the average person, or sorry, at 20 feet, you can see what the average person could see at 15 feet. So 20/15 would be better than normal vision. Hopefully that all just made sense. So here's our vision. Again, this is showing us the refractions. So emmetropia down here means normal vision. So we have light coming in, getting refracted at the cornea in this direction, and then getting refracted again at the lens and converging onto the focal point. Nearsightedness or myopia is where the eyeball is either too deep or the resting curvature of the lens is too great. So what happens here is the light gets refracted and focuses in front of the retina. So that needs to be corrected so that the light doesn't converge too close, so it helps to spread out the light. So you can see down here with this diverging lens, and I'm not going to test you. I'm never going to ask you what kind of a lens do you use to correct for this. That is beyond the scope of undergraduate A&P 1, but I think it's cool to see, and many people wear glasses, so it's interesting to learn about that this lens, this diverging lens, takes these light rays here and spreads them out prior to them hitting the cornea and then being refracted again. So it's a diverging lens, so it helps to center the vision back on the appropriate focal point on the retina. Hyperopia is farsightedness, and so it's the exact opposite. So either your eyeball is too shallow or the lens is too flat, so it's not refracting the light enough. So by the time the light all comes together and focuses, that focal point is behind the retina. So to correct for hyperopia, you need a converging lens, which is going to bring these down here on the bottom. It's going to bring the light rays closer together prior to hitting the lens or prior to hitting the cornea, and then it's going to allow you to properly refract that light to focus a clear image onto your retina. One of the other things that's important to know about with vision is that when we are looking at things, the vision is actually created upside down and backwards. So this is showing you that if you're looking at an object here, the light from the top of the object is going to be transmitted to the bottom of your eyeball, and light from the bottom of the object will get transmitted up to the top of your eyeball. So that image is arriving upside down. So here's the bottom to the top. So here we're looking at a telephone pole. So you can see that the telephone pole, you are seeing it upside down here. And then you look at it backwards, and we'll see that we'll look at a fence to look at how we see things backwards. So if we're looking at this fence, from the red on the, we'll say the right side of the fence as you're looking at it, is actually going to cross over and show up on the left side of your inner eye. And the left side of the fence is going to cross over, and that image will show up on the right side of your eye. So you're looking at this fence backwards. And then you can also, if you look closely at this picture, you can see that the tips of the fence posts are pointed and the bases are square. And so over here, those pointed tips are upside down and the bases are on top. So the images that you are seeing are upside down and backwards. And then they get processed within your brain to, again, show you more of a right side up image. I am attempting to erase all of this. There we go. So earlier we talked about the photoreceptors being rods and cones. And those photoreceptors allow for two different visual systems, photopic vision and scotopic vision. Photopic vision is rod-based, so it is highly light sensitive but not color sensitive. So remember, rods are for utilizing in dim light versus cones, which are for color vision. So our photopic vision is based on the rods, so it supports your vision when the light is low. The rods are cylindrical cells on the outer layer of the retina. They provide peripheral vision and vision at night. Scotopic vision is less light sensitive, but it's color sensitive, made up of your cone cells. So the cone cells absorb light from three different portions of our spectrum, which is red, greens, and blues. Cones are a specialized type of neuron responsible for color perception. They function best in bright light, so cones don't function very well in dim light. In dim light, your rods take over and you see more of a grayscale vision in dim light. But in bright light, we see cones, and we use our cones to see multiple different colors. And they allow the perception of color vision and finer detail, so that visual acuity is more of a cone-based vision. So rods and cones of the retina are our photoreceptors. They're detecting photons, which are the basic units of light. Light energy is a form of radiant energy, and it travels in wavelengths. And we're not going to get super deep into the physics of light, but just to know that the human eyes are sensitive at wavelengths of 700 to 400 nanometers, which is the spectrum of visible light. And you can use the acronym Roy G. Biv to know what colors we can distinguish, which are red, orange, yellow, green, blue, indigo, and violet. And there's a fun little video you can watch about that. And here's a picture of those different wavelengths and the colors that they involve. Blue is the shortest, or violet is the shortest of the wavelengths. Red is the longest of the wavelengths, and the rest of them fall somewhat in between as far as the cones go. And then you can see that those rods are working best between at the 500 to 550 nanometer of wavelength. Now there's a very detailed portion in your book about the physiology of vision, and we are not going to cover that whole section. There's a lot of information there, and I feel like it's too much to really do justice in 1 week. So there's just these small bits of information that I want you to retain from that section. The rods and cones have visual pigments which are absorbing the different photons of light. Rhodopsin is the visual pigment found in rods, and it contains opsin bound to retinol. Retinol is a vitamin A derivative, so when people tell you to eat carrots to improve your vision, that actually is a thing. So the retinol is derived from vitamin A. Vitamin A is found in carotene, and so eating carrots can actually help your vision. We have blue cones, green cones, and red cones, and each of those cones responds to a different wavelength of light, which is what is going to help us distinguish one color from another. When all of the rods alone are stimulated, or when all 3 types of cones are stimulated at the same time, then you're going to be perceiving white light. Photoreception is our sensitivity to light. So how sensitive are you to light? And the one thing that I want you to read on page 588, this is an old edition, so on this newest edition, it's page 588, is about bleaching and regeneration. So basically what happens is rhodopsin gets broken down and reassembled within your the rods. And when all of the rhodopsin is broken down at once, that's called bleaching. So it means that you can't, nothing can change because there's no more photoreceptors to stimulate, right? All of the pigment is utilized and needs to be reassembled. So during bleaching, our photoreceptors are inactive, or during regeneration, they're bleached, they're regenerating, the photoreceptors are inactive. Bright light can produce a ghost image through those photoreceptors, which is pretty cool. So if you've ever gone out into the sun and then you close your eyes and you can still see the sun, that's because of this bleaching. Your body contains enough vitamin A reserve to produce visual pigments for several months. So I thought that was a cool fact, that even if you're not getting vitamin A on a regular basis, you can still produce it for multiple months after you haven't, without consuming it. This is a picture from the Ishihara book for color blindness testing. On your handout, if you printed this, you're not going to be able to see anything because it is gray and red, but this is showing you the number 12. If you're watching the video, hopefully you can see that. Color blindness occurs when one or more types of the cones are non-functional. The most common color blindness is red-green color blindness. And so when somebody with color blindness looks at this book, they can always see the first page, the first number, and then when you turn the page, they usually look at the second 2 numbers and they're like, I don't know. So it's pretty interesting. So men, 10% of men have some sort of color blindness versus 0.67% of females. So it's much more of a male dominant issue to be colorblind. So now we're going to talk about the visual pathway. So what happens, we talked about how do you form an impulse basically within the ganglion cells that are going to send their axons into the optic nerve. So we know how that happens. So what happens once those ganglion cells are stimulated? So the optic nerve, cranial nerve 2, transmits visual information from the retina to the brain. It's encased within meninges and contains the axons of those ganglion cells. Ultimately, it's going to terminate in the lateral geniculate nucleus of the thalamus. Visual processing, reflexive eye movements, and sleep-wake cycle functions are affected by the transmission of light. The second synapse allows the visual information to begin to integrate and process within the retina before reaching the central nervous system. So the fact that the photoreceptors are going to be synapsing on the bipolar cells, and then the bipolar cells are synapsing on the ganglion cells, and we have those horizontal and amacrine cells, we begin to process all of that information in the retina. So the information that's leaving the eye in the optic nerve is already undergoing preliminary processing before it even gets sent to the thalamus. So that's basically what this slide is saying. A little throwback again to A&P1, the idea of convergence. We were talking about neuronal circuits and how you can have divergence where you start with a synapse at one cell and then it goes out to many different cells, or convergence where you have information from a whole bunch of different cells coming down and all converging onto one cell. So each ganglion cell, we've got M cells and P cells, are two different types of ganglion cells. The M cells, each M cell gets information from up to 1,000 different rods. So the rods are going to converge onto a ganglion cell. So you can have 1,000 rods converging their information onto one single ganglion cell, and that's going to be an M cell. So the M cells are monitoring the rods. The P cells, the P ganglion cells, are monitoring the cones, and there is no convergence in that system. So the cones, it's one cone to one ganglion cell, versus the rods can be up to 1,000 rods onto one ganglion cell. So I thought that was an interesting tidbit as well. All right, so visual pathway. This is interesting because your eyes are set apart. And so you've got vision from the lateral side of each eye and the medial side of each eye. And that vision is going to get sent. So here's your eye. Comes down the optic nerve to what's called the optic chiasm, is where your two optic nerves sort of come together. And then the information splits and goes back to this lateral geniculate nucleus. So information, your vision kind of splits in half at each eyeball. The vision from the lateral half of each eyeball, so this information, is going to come down and stay ipsilateral, like that. The vision from the medial half of each eyeball comes down the optic nerve and then crosses over or decussates to the opposite side of the brain. So we can look at lesions in this system in multiple different places and determine what kind of loss of visual losses we could have. So if somebody presents with strange visual loss, you could figure out where in the system the lesion would occur. So let's say we have a lesion here. So in the right optic nerve, we've got a lesion. So that lesion of the right optic nerve is going to create total blindness in the right eye because you've got the medial and lateral portions of the right eye are sending information through that right optic nerve. Similarly, if it was through the left optic nerve, you would have total loss of vision in the left eye. Now, if we had a lesion in the distal to the optic chiasm, I'm trying to do a different color here. So if we had a lesion here, now we would lose lateral vision from the right eye, this red here, and we would lose medial vision from the left eye. You see how that works? So it's really interesting how you can lose different portions of your vision depending on where those lesions occur. You can also see on here that from the lateral geniculate nucleus, information gets sent to the superior colliculus. That's where you've got those reflexive vision. You see something on the side of your vision and you reflexively turn to look at it. That's because you have information from the optic nerve coming into the superior colliculus. And then you also have information ascending from the lateral geniculate body up into the diencephalon and the brainstem, and projecting to the visual cortex of the occipital lobe. All right, so that finishes vision and the eyeball. And the last thing we have to do is the ear. The ear is divided into 3 sections. We're going to look at the anatomy of the ear, and then we're going to look at the physiology. And the ear is responsible for hearing, but it is also responsible for balance and equilibrium. So the ear itself is divided into the outer ear, the middle ear, and the inner ear. The external ear, or the outer ear, has 2 parts. The auricle, which is what we think of when we say your ear, like did you get your ear pierced? That would be your auricle, which is also called your pinna. And then the external auditory canal, which is basically the hole that enters from the auricle and ends at the tympanic membrane. The acoustic function of the ear is to transmit sound from the external environment to the tympanic membrane. And then non-acoustically, the ear is self-cleaning, so it produces cerumen, which is earwax, and the cerumen is keeps you, it's antibacterial, and it also helps to keep like little bugs and things like that from getting in your ear, and it helps to move objects away from your eardrum. So that's basically what I was just saying. This wax shuttle is where that cerumen is moving away from the tympanic membrane, which is the eardrum, at all times, trying to protect your eardrum. If you have too much earwax in your ear and it gets impacted, it can actually interfere with your ability to hear. When we're looking at how sound gets into the eardrum, the auricle, or the pinna, actually helps to adjust the sound and bring it toward the eardrum. And then the canal amplifies the sound, which is going to make more of an impact onto the tympanic membrane. So the auricle is what helps you to localize sounds in space. Like you can know if a sound is coming from behind you or in front of you or to the right or left of you if your eyes are closed. And then the actual canal itself is going to amplify that sound. The middle ear is made up of the tympanic cavity, and that is an air-filled cavity. The air comes through the nasopharynx, or from the nasopharynx through the auditory tube, which is also called the eustachian tube. And it contains, the middle ear contains our ossicles, which are the 3 smallest bones in our bodies, called the malleus, the incus, and the stapes. And there are little tiny synovial joints between each of those bones. So those are the smallest synovial joints in our bodies. The purpose of the ossicles is to transmit the vibration of the tympanic membrane through those bones to the inner ear, which is where the stapes, which is this one here, is covering another membrane called the oval window. So the ossicles are transmitting vibration from the tympanic membrane across the middle ear to the inner ear. So the boundaries of the middle ear are the tympanic membrane is the lateral boundary, and then the oval window is the medial boundary. The tympanic membrane itself, which is also called the eardrum, is a thin, semi-translucent, so it's like a pearly color, like that mother of pearl. So you can see through it. When you look in somebody's ear, you can see through it and you can see the bones on the other side, but it's not totally clear like cellophane paper. It is concave shape on the outer surface because of the bones kind of pulling on it, pulling medially on it, I guess. The function of the tympanic membrane is to transmit the sound waves that are coming in the external canal. It transmits those waves to the bones of the middle ear, which then transmit it into the inner ear. So sound waves are going to reach the eardrum. The eardrum or the tympanic membrane is going to start to vibrate along the same resonance as the sound waves that have come in, and then the movement of the tympanic membrane is going to move or vibrate those little ossicles, which will move the sound toward the inner ear. Trauma to the tympanic membrane can happen if you were to stick a foreign object in your ear. You could perforate the membrane. External pressure, so like an explosion, a very loud sound. With an explosion, you've got more of that actual boom part of it, so it's more of the pressure of the boom. Or if you were to slap somebody really hard on their ear, that could create a pressure that could actually perforate or rupture that tympanic membrane. It can also rupture from internal pressure, so a middle ear infection could have pus or exudate that could be pressing on the tympanic membrane and allow for a rupture of something coming out of the membrane. Result would be scar tissue of the tympanic membrane. Scar tissue is not going to be as flexible as the original tissue was, so that decreased flexibility can over time interrupt the ability of the tympanic membrane to conduct sound appropriately, and you can end up with what's called a conductive hearing loss. So that's why kids who have lots of recurrent ear infections and maybe a rupture are much more prone to having tubes put in their ears so that when you put a tube in, you have a very small amount of scar tissue. When you rupture a tympanic membrane, you end up with a lot more scar tissue. So that's why I ended up having tubes put in my little one's ears, because she ruptured her right tympanic membrane twice and her left tympanic membrane once with her ear infections, and so she has a lot of scar tissue in her ears now, which thus far, knock on wood, has not interfered with her hearing. The ossicles are those little bones within the inner or the middle ear. So they are the malleus, which is hammer shaped, the incus, which is supposed to be shaped like an anvil, and the stapes, which is shaped like a stirrup. You can see the stapes really clearly right here. The malleus is in direct contact with the tympanic membrane. The incus is in the middle, and then the stapes is covering the oval window. So vibration is going to go from the tympanic membrane through the malleus, the incus, the stapes, and then the round or the oval window. The tympanic membrane is going to convert sound waves into mechanical movement, and that's going to move those ossicles and then vibrate over the oval window, transmitting into the inner ear, which is actually full of fluid. We do have an acoustic reflex, which is a protective mechanism that if there is a very loud sound, the tensor tympani muscle can tighten and stiffen the tympanic membrane so it doesn't allow it to move as much as it would, and that can therefore be protective. The stapedius muscle also reduces the movement of the stapes bone at the oval window to protect the inner ear. But that only works, we'll look at the next slide. So it doesn't happen quickly, and it's only effective in cases of not too sudden and not too terribly loud or not too prolonged. So basically, if you are, working with a jackhammer all day long, or you are an explosive expert and you don't wear ear protection, this is not going to help you. But if you're, if your neighbor sets off an explosion, like sometimes we're that neighbor and we like to blow stuff up in our yard, then this reflex can help out our neighbors who are kind of far away, so it's not right in their face. So that makes me feel better about blowing stuff up in my yard, which is super fun. The eustachian tube, which is also called the auditory tube or the pharyngotympanic tube, connects the middle ear cavity with the nasopharynx, which is in that sort of upper throat palate area. This is the tube that is pretty horizontal when you're a little kid, and it becomes more vertical, more of like a 45-degree angle in an adult. So it has the ability to for bacteria and viruses from the nasal cavity to move up the eustachian tube and cause infections of the middle ear. But its main function is to drain the middle ear if there's anything in there, and then to act as that pressure release valve to help you normalize the pressure between your middle ear and the atmospheric pressure. So yawning and swallowing open the eustachian tube and allow for you to normalize that pressure. If the eustachian tube gets blocked, and you end up with a buildup of fluid and mucus in the middle ear where it doesn't belong, that can also decrease your ability to hear. The last portion of the ear is the inner ear, and part of it is for hearing and part of it is for balance and equilibrium. So the inner ear contains a bony labyrinth, and inside the bony labyrinth is a membranous labyrinth. The membranous labyrinth is full of a thick fluid called endolymph, and then the membranous labyrinth is surrounded by perilymph and then contained within that bony labyrinth. The regions of the inner ear are the vestibule, the semicircular canals, and the cochlea. The vestibular labyrinth is responsible for balance and equilibrium, and that's going to be this whole portion back here. And then the cochlea is responsible for hearing, and that's going to be this snail-shaped portion here. So the semicircular canals make up the vestibular labyrinth, and it's 3 canals that lie at right angles to one another, so they represent each plane of a 3-dimensional space. So we can move in 3 dimensions, we live in 3 dimensions, so each of these canals represents one of those 3 planes. The vestibule is the center area where those 3 canals come together, and that's also where the cochlea begins. And all of those structures work together to help achieve balance and equilibrium. And they're going to respond to changes in gravity as well as directional movements of the head. So you can turn your head, you can tilt your head, you can speed up, slow down. All of those things are going to be determined by the movements of the fluid within these semicircular canals. So our sense of gravity and acceleration is through the hair cells that are within the vestibule. So remember, the vestibule is where the 3 canals come together, So you can see here in step 1, his head is in this neutral position, and you've got these otoliths. Otoliths are like little stones that are within the vestibule. And so those otoliths are sitting on top of hair cells. And these hair cells are neurons. Excuse me one second. Okay, I'm back. Sorry about that. So the otoliths are these little stones that are sitting on the hair cells, and the hair cells are like little neural receptors. So when you're at rest or in neutral position, these stones are pressing straight down onto these hair cells. So the hair cells are sort of turned off. They're used to having that pressure. But when you tilt your head backwards, like in step 2, you can see that those stones start to move off of those hair cells, and that triggers the hair cells to send signals to say, hey, we're tilted backwards. Or if you tilt your head forward, those stones would move in the opposite direction, and you would know, hey, we're tilted forward. Or if you're accelerating, they're going to move backwards, and if you're decelerating, they're going to move forwards. So that's how the hair cells within the vestibule are what tell you about your gravitational position. So like if you're in anti-gravity, then the stones, I guess, would not even be touching the hair cells. So then they would be stimulating to know that you're not touching the hair cells, as well as the fact that you'd be floating in the air. But if you are accelerating or decelerating or tilting your head forward or backwards, then those hair cells will be stimulated accordingly. Rotation, a sense of rotation, is going to be modified or transmitted by the semicircular canals. So the impulses are sent to the cerebellum through the vestibulocochlear nerve. Cranial nerve 8 is the one that is going to be carrying information about balance and equilibrium, as well as information about hearing. So head position is going to go through the vestibular portion of the vestibulocochlear nerve and go to the cerebellum, and then the cerebellum will trigger signals back to the skeletal muscles to make you adjust your positions accordingly. If you want to know what each of the canals does, the lateral semicircular canal is going to register if you say no. So if you rotate your head from side to side, saying no is the lateral canal. Saying yes is the anterior canal, and tilting your head to either side is the posterior canal. So now we're going to move on to the cochlea, and the cochlea is the auditory portion. So the end of the hearing process that we've been looking at is what the cochlea is involved in. So the cochlea is embedded within the spongy area of the mastoid bone, just behind the hinge of the jaw. It begins at the oval window. If you remember that the stapes was contacting the oval window. And then it curves into that shell looking, looks like a snail shell, and then it ends at the round window. So the oval window is the membrane against the stapes, which is going to transmit information into the inner ear and set the fluids of the cochlea in motion. The oval window is collagen fibers. It's connected to the stapes, and it's receiving the vibrations from the middle ear. There's perilymph within the cochlea, so that window is going to take the vibration of the bone and change it into a vibration within the fluid of the cochlea. And then that vibration is going to move through the cochlea, stimulating the cells within the cochlea, and then it's going to come up against the round window at the end, which is another membrane, And basically what the round window does is it provides a counter pressure to the pressure that's coming in at the oval window. So the pressure needs a place to kind of go. So instead of just sending pressure continuously into the cochlea, it's going to then bounce against the round window to kind of counterbalance that pressure. Both of those structures are going to work together to create pressure waves within the perilymph that's in the cochlea. The cochlea is made up of 3 fluid-filled canals. We're going to start with number 2. So the scala vestibuli is the first canal that is up against the oval window, and it lies above the central canal. The scala media, which is the central canal, is going to be in the center. And it contains so that's number 1 up here, which is also called the cochlear duct. So it contains endolymph. The cochlear duct contains endolymph. The scala vestibuli and scala tympani contain perilymph. So the cochlear duct contains the spiral organ, which is also called the organ of Corti, C-O-R-T-I. And that's the main component of hearing. It's got little hair cells in it, and the hair cells are stimulated by the movement of the endolymph across the hair cells themselves. And then the scala tympani contains perilymph and runs up against the round window. So the process of hearing, if we start from the beginning, the sound comes in the external auditory canal. It creates resonance in the tympanic membrane. The movement of the tympanic membrane is going to move the 3 little bones in the middle ear. The stapes, the movement of the stapes on the oval window is going to start moving the perilymph and the scala vestibuli. That's going to then create movement in the endolymph of the cochlear duct, which is also called the scala media. And in that movement of the endolymph is going to stimulate the organ of Corti, or the hair cells in the spiral organ, to depolarize. And then from there, the scala tympani, the perilymph within the scala tympani, is going to move up against the round window to sort of equal out that pressure that came in from the oval window. So there's a nice little video here that you can watch about that. The auditory pathway, beginning at the cochlea, we've got the cochlear portion of the vestibulocochlear nerve. It's going to come and synapse in the cochlear nucleus, and then from there we'll go up into the inferior colliculus, and that's how that auditory reflex turning toward a noise that you hear, the auditory reflex is from the inferior colliculus. And then from the inferior colliculus, it is going to move up into the thalamus, and then from the thalamus, it'll get projected to the auditory cortex in the temporal lobe. So that is the auditory pathway. Our hearing range is the range from the softest power that we can hear up to the absolute loudest sound. We never use our full potential because of that reflexive protective mechanism. Young children have the greatest range of hearing. They hear between as low as 20 hertz and as high as 20,000 hertz, which is pretty incredible. To give you an idea of different noise levels, we're looking at decibels. That's what we measure noises in. So the lowest audible sound is 0 decibels, which most people who are not little children cannot hear at 0 decibels. Soft whispering in a library is 30 decibels. Light traffic in the distance is 50 decibels. A noisy restaurant is 70 decibels. Heavy metal, like going to a rock concert or being at like sandblasting, is 120 decibels. Being at a rocket launching pad is 160 decibels. And you can see here at 160 decibels, hearing loss is inevitable if you don't have hearing protection. So this is telling you when dangerous exposure is for each of those different decibels and how likely you are to damage your hearing. So a little plug here for always wear hearing protection if you're doing anything loud. It is not worth losing your hearing. As we age, as with all other systems, things start to go wrong. Damage from all the years of doing stupid things starts to accumulate. The tympanic membrane becomes less flexible, so it's less able to vibrate at the same resonance that it could earlier. Articulations between those little ossicles begin to stiffen, so they don't resonate as much as they did. The round window can also begin to ossify, so it doesn't balance out that pressure as much. So all of those things together can create hearing loss over time. So this slide and the next one are just showing us from beginning to end how hearing occurs. So step one, we've got sound waves that are arriving at the tympanic membrane, and you can see that here. Step two, the tympanic membrane is going to begin to move, and it's going to cause displacement of those auditory ossicles. So that's happening here. Step three, the movement of the stapes at the oval window is going to establish pressure waves within the perilymph of the vestibular duct. And so that is occurring here. So we've got these pressure waves. So right here, we've gone from the outer ear here to the middle ear here to the inner ear. All right, so now we're moving the fluid within the inner ear. Step four, the pressure waves are going to distort the basilar membrane on their way to the round window of the tympanic duct. The vibration of that basilar membrane is going to vibrate those hair cells against what's called the tectorial membrane. I'm not going to ask you the difference between those membranes, so just know that the vibration is going to stimulate those hair cells, and the information from the hair cells is going to be relayed through the central nervous system through the cochlear branch of the vestibulocochlear nerve. Here's some videos you can watch about conductive hearing loss versus sensory neural hearing loss. Conductive hearing loss would be damage to the outer, middle, or inner ear structures. Sensory neural hearing loss is damage to those hair cells or the auditory tract within, the vestibulocochlear nerve. Strong movement of the endolymph and the spiral organ from a really loud noise can cause those hair cells to die. That's a pretty common reason for partial hearing loss. So people who are repeatedly shooting guns without hearing protection, or blowing stuff up, or going to tons of concerts when they're young, all of those things add up and cause damage to those hair cells and can lead to hearing loss later on. Any impairment of the spiral organ, so those are the hair cells themselves, or its connection to the vestibulocochlear nerve, can cause that sensory neural hearing loss. So when it's the nerves that are damaged, they are not going to regenerate, and you're not going to get that back. And it's harder to correct for sensory neural hearing loss because it's the nerves that are actually not transmitting the impulses, versus conductive hearing loss, where if you say the tympanic membrane is stiffer, if you put in a hearing aid and you amplify those sounds, then you can create additional movement of the tympanic membrane. So conductive hearing loss is easier to correct for than sensory neural hearing loss. Other causes of sensory neural hearing loss are a disease called Meniere's disease. You can have congenital sensory neural hearing loss, environmental noise exposure, systemic diseases, aging, or drug toxicity. So that is the end of our extremely long lecture. I super apologize for that, but there's so much information in this chapter to get through. I wanted to make sure that you were able to hear me explain it all if it be your pleasure. So next week, I can't even remember what we're doing next week. How exciting is that? Let's see. Next week, I don't even have it in front of me. Next week is a mystery. So I will see you next week, and I hope you have a wonderful week.